Hey, hello, good morning from Michigan. I'm pleased to welcome all of you to the ZOOMinar on proteinopathies. Let me begin with a very important announcement. The panel has put together a fantastic plan to better involve the participation from early career researchers like students and postdocs in your lab or throughout the world. It's a pity that we didn't have any conferences for you to go and participate for almost a year now. But we are bringing this opportunity to you to exhibit your exciting recent research findings. All you need to do is to prepare an abstract and a short video. No need to travel, and believe me, no registration fee and no hidden fees as well. A committee will then select many contributions for live ZOOMinar presentations. Guess what? We have sponsors lined up already and more to come, and we'll definitely award several presentations. More details can be found in the symposium website. You know the website, so it has been sent out by email as well. My request to all mentors, faculty, and colleagues, please encourage your cohorts from your group and collaborators groups to participate in this fantastic outreach activity. Okay, we have a fantastic speaker today, Dr. Elizabeth Rhoades from UPenn. Liz is alumni, Michigan alumni. So she's special to us. So for a special person, we have a special introduction by my colleagues from Michigan Biophysics, Professor Ari Gaffney. Ari needs no introduction to this audience. He has made excellent contributions to protein folding research. Thanks, Ari, and Duncan and Gillard for coming. Thanks, Ari, for introducing the speaker. Go ahead. Ari, you need to unmute yourself.
I did. Well, it's really my pleasure to introduce Liz Rhoades. Liz and I met many years ago when she came to biophysics as a young grad student. Decided to work with me and with Duncan Steele, who is also here. The work involved a small peptide, IAPP, which is involved in type 2 diabetes, and which is known to aggregate, much like the amyloid peptide of Alzheimer's disease does. And we worked on it for about 4 or 5 years. Liz got her PhD. She discovered a very interesting and new feature of this peptide. It tends to aggregate to form micelles before aggregating further to form the amyloid type fibrils. And these amyloid micelles, these small aggregates, may be toxic to cells. Liz invites me in Israel to work with Professor Gilad Aran as a postdoc, where she got introduced to a single molecule spectroscopy and detection. And the work involved protein folding. And she did some beautiful work over there, came back to the States and postdocs for about a year, I think, with Watt Webb at Cornell, and then moved to Yale, where she got her first tenure track position. She rose through the ranks there and finally moved to UPenn about 5 years ago now, something like that, where she is now focusing her work on small proteins that's also involved in Alzheimer's disease and other Parkinson's disease, alpha-synuclein, and her work on alpha-synuclein is the subject of her presentation today. Liz was really a super student. She's a great scientist, and I'm really looking forward. We haven't kept in touch for the last year because we haven't seen each other because of the virus, and so I'm really eager to hear what she's doing these days. Liz, please.
Okay, so I'm so happy to be here. I'm thanking the organizers for inviting me, and then also it's wonderful to have so many people from my scientific upbringing here and to have that wonderful introduction by Ari. So I'm actually going to turn my video off just because we have bandwidth issues at my house sometimes. So I'll turn it back on for for questions, but I'll have it off for the duration of the talk. Okay, so with that wonderful introduction, I will tell you the primary story I'm going to tell you about today is a story about alpha-synuclein. But I'm going to start with a broader introduction to the, well, first of all, by introducing my lab, sorry. So this is a picture we took this fall, a socially distanced group picture that we took this fall in celebration for one of my students, Jenny here, who just, well, actually in the spring had completed her qualifying exam, and we finally got a chance to celebrate at the end of summer. Almost all the work I'm going to tell you about today is a work of a fantastic postdoc, Melissa Birold, who has just started an independent position at the Max Delbrück Institute in Berlin. But she's really the architect of all of this work, although it builds upon some past work from other team members, of course. So I'm just going to introduce to you, because we're generally interested in my lab, we're interested in the class of proteins that is known as intrinsically disordered proteins. So these are proteins that lack stable secondary and tertiary structure. And last week, of course, you heard a lot about tau in Mei Hong's beautiful talk and the structural work they've done with it. Tau is another one of the proteins that we study in my lab. And what it shares in common with alpha-synuclein is that it's completely disordered in solution, forms, of course, the structured fibers when it aggregates, but that it also interacts with a variety of binding partners. And while we know less about alpha-synuclein's binding partners, we've known for decades that tau is a microtubule binding protein. And in fact, this is a cartoon of tau here. And so the major domains you see are the N-terminal domain, this proline-rich region, the microtubule binding domain, so named because it's thought to contain the primary microtubule binding sites. It's also have been heavily studied because the minimal sequences for aggregation are found in R2 and R3 of this domain, and then the C-terminal domain. And there's a lot of different splice isoforms of tau, and those are the either spliced down here in the N-terminus or splicing of this R2 domain. So you also saw, I think last week May mentioned this structure from the Nogales group, which was sort of a technical feat in that they were able to actually resolve tau bound along the surface of a microtubule. And you can sort of see that they looked at primarily this R2 and R1 domains, and you can see that it stretches out across the microtubule lattice. And we were also interested in how tau binds to microtubules and tubulin. And the reason I'm talking about this is because I think it illustrates the challenges we have in sort of understanding function of these disordered proteins. Because despite the fact that since tau was first identified as a microtubule protein by Mark Kirshner some 40 years ago, there's still a lot of open questions about how it functions. And so this is recent work from a student, Kristen, in my lab, where she looked at both this proline-rich region. So this is a somewhat, I guess, mysterious region of the protein. It's known to be important for binding protein. The reason we care a lot about it is because almost all of the phosphorylation sites that you frequently hear about being linked to Alzheimer's disease and other tauopathies are found in this proline-rich region. And yet we understand very little about what its actual role is. So Kristen was interested in the proline-rich region. And so she did some comparative studies of fragments of the protein where either it was just the proline-rich region or the proline-rich region and the flanking microtubule binding region. And so these are some results from her work where she was looking at these constructs binding to soluble tubulin, so not microtubules, but soluble tubulin dimers by FCS. And so what you're looking at here is an increasing amount of tubulin as we go on here. And what you're looking on the y-axis is the normalized diffusion time. So this is how fast something is diffusing. So the higher the number gets, the bigger it is. And I've drawn a little cartoon up here just to illustrate that for us. We have our tau construct labeled, so it has a fluorescent tag on it. The diffusion time of the construct alone is going to be faster than the diffusion time of the construct bound to tubulin, which itself is going to be faster than when the construct is bound to multiple tubulin dimers. So if we look at the PRR, you'll see something quite interesting, which is that it binds tubulin all on its own in the absence of the microtubule binding repeats. And you actually, the curve here is not great because the axis is not ideal, but what we can see if you blow this up is you actually have two phases of binding. So this is our first phase where we have no tubulin bound. We have a step in the binding which corresponds to a single tubulin dimer bound, and then it saturates up here with two tubulin dimers bound. And then what we want you to do is contrast that to what happens when we have the proline-rich region and the microtubule binding repeat present. You can see that at low concentrations of tubulin, these curves, these diffusion times more or less overlie each other, suggesting that you're having that stoichiometric one-to-one complex binding. But But then as we continue to increase tubulin, this longer construct, in contrast to our shorter construct, starts to form these things with very large diffusion time. So these very heterogeneous assemblies, which are, many, many tubulin dimers bound. And this is where you get the deviation. And so based on that, we hypothesize that actually the proline-rich region might serve as a primary binding site for the tubulin dimer, and that this, in the context of the function of helping to polymerize microtubules, this microtubule binding region serves to help increase the local concentration by binding multiple tubulin dimers, and this is how it goes about in facilitating polymerization. And I will point out that this PRR alone is capable of forming microtubules. So this is actually TEM of microtubules formed in the presence of the proline-rich region. So not only is it capable of binding tubulin, it's also capable of polymerizing this into microtubules. And so this is, I think, a nice illustration of what we do not understand about functions of disordered proteins, because as I said, we've been studying tau as a microtubule binding protein for 40 years. And the fact that the PRR is capable of independent polymerization was something that we just discovered last year. So there's still a lot that goes into understanding the disordered proteins. And this is made even more complicated in the case of alpha-synuclein, where we don't actually even have a clear understanding of who its functional binding partners are. So this is my cartoon of alpha-synuclein. So it's smaller than tau. It's only about 140 residues long. Its aggregation is linked to Parkinson's disease, as I'm sure everybody here knows. I'm illustrating the three main domains for those of you who are not as familiar with alpha-synuclein. Roughly the first 95 amino acids or so form a membrane binding domain, where it forms an amphipathic helix upon binding to anionic vesicles. It has this central NAC region, which is the most hydrophobic part of the protein. This is also the core, forms the core of fibers when alpha-synuclein aggregates. And then it has a highly acidic C-terminus carrying a charge of, I think, -13 over these 40 residues on the C-terminus. So it's very negatively charged. There are a number of point mutations that are linked to early onset familial forms of Parkinson's disease. These are all found down here in the membrane binding domain. And actually there's I think I'm missing an A53V also. So there's probably others that are gonna be discovered over time. I've already said we don't really know what alpha-synuclein does because of this membrane binding domain and some functional studies. It's thought to involve maybe interactions with synaptic vesicle trafficking and regulation, or have a role in maintenance of neuronal plasticity. This is from the very early functional studies from Julia George and colleagues, where she was looking at learning, birdsong learning in model systems, and found that synuclein seemed to have a role in sort of timing of when bird songs were learned. And there's been other animal studies which show that you get some changes in synaptic structure or deficits in recycling when you have a loss of alpha-synuclein. And then of course the aggregation is implicated in Parkinson's disease. So like most disordered proteins, alpha-synuclein is subject to a lot of post-translational modifications. So I already alluded to, for example, the phosphorylations that are found in tau. Synuclein is also similarly modified by all sorts of modifications, including side chain phosphorylation, ubiquitination, glycation. But the one I'm going to focus on is actually this N-terminal modification, which is acetylation, because it's somewhat different than all the others. And what I mean by that is most of these other regulations that we're talking about are regulatory modifications, right? So when we think about phosphorylation, right, it's a reversible interaction. It will get regulated as a function of what's needed from the function of the protein. And N-terminal acetylation is different from most side chain modifications because it occurs co-translationally. So in the case of the enzyme that modifies alpha-synuclein, that's NATB, it's associated with the ribosome. So sort of as soon as the chain is long enough for the NATB to access it, you'll have acetylation of the N-terminus. And it's not reversible. And what I mean by that is that currently there are no known N-terminal deacetylases, so no enzymes that will deacetylate the N-terminus. And that's in contrast, for example, to side chain acetylases. For lysine side chains, there are enzymes that will deacetylate those side chains, and this is not true for the N-terminus. So it's a really common modification, depending on how recent the review you read is, maybe as many as 80% of all mammalian proteins are modified by N-terminal acetylation. And from what the description I've just said, we can describe it as constitutive, meaning that once the modification is there, then it doesn't get reversed. And for a lot of proteins, it's critical to recognition of binding partners. A good example of this, or a canonical example of this, is the protein tropomyosin. So this is the actin binding protein. And one of the reasons that it's been purified from animal sources for so long is because in the absence of that N-terminal acetyl group, it doesn't really bind to actin that well. And in fact, we were very fortunate in that there was a group who was working on figuring out ways to make tropomyosin with that N-terminal acetyl group in E. coli, and developed the NATB system that allowed them to do that. And we got a hold of that plasmid, and that's what allowed us to also make alpha-synuclein that was N-terminally acetylated in E. coli, which is sort of the crux of some of the results that we find. All right, so this is actually, if you look at the date of this is work that's almost a decade old now. And it's basically an observation we made, which is, this is showing you protein. So I said we were able to get this in the plasmid from the Mulvihill group and then co-express the NATB complex with alpha-synuclein. And it actually works beautifully in terms of allowing you to make the acetylated protein. And we were interested in the impact of the acetylation on alpha-synuclein's interactions with membranes primarily, but as part of that, we were purifying the protein by a couple of different methods. So the first one was this ammonium sulfate precipitation, which is a standard method for alpha-synuclein purification. And the other was a milder protocol, which we called our BOG method, because it used the detergent beta-octoglucoside and glucose. And what you'll see is if we took the unmodified protein and purified it and ran CD on it, you get a spectrum that looks more or less like an unstructured protein with that negative peak down below 200. But if we took the acetylated protein and purified it by the same two approaches, we see that the protein purified by BOG looks quite different. And I will note, we this was after extensive dialysis, so we'd done as much as we could to try and remove all the BOG, and clearly we were successful over here with the unmodified protein, but not as successful with the acetylated protein. So that suggested to us that the acetylation had somehow conferred a unique interaction for alpha-synuclein with BOG. And this is just what BOG looks like. So it's a carbon chain with that glucose group on the end of it. And that had us asking, so what's unique about BOG compared to, say, other detergents that we might have used? And so if we look at SDS, which we've also used for purification, you can see the major difference is rather than having this, this charged head group, we instead have a sugar group. And to us, this was striking because we know that there are a lot of lipids that are modified by sugar groups in neurons, right? So, as many as 5%, I think, of the lipids that are found in neurons are modified by sugar groups. And so that had us very interested in whether or not there was some unique interaction between the sugar groups found on these lipids and the acetyl modification on the N-terminus. So I've already mentioned this is, we know that synuclein binds to anionic lipid vesicles. This is something that has been studied for years and years, not only by us, but by many other groups. This is a cartoon from some very nice work by Ashok Deniz, which emphasizes the fact that even when it's membrane bound, synuclein can take on multiple different conformations. So this is showing a bent helical conformation when it's bound to relatively small detergent micelles, and then this elongated helix that forms on more planar membranes. So this has been known for a long time, but the majority of the studies that have been done on alpha-synuclein have been done on, including ours, have been done on synthetic lipid vesicles. So these were model systems that were relatively simple compared to the cell membranes where synuclein would normally be found. And this is just my cartoon to remind us how complex cell membranes are. Depending on which side of the membrane you're on, you're going to see different protein groups. You're going to see, if you're extracellular in particular, you're going to see a lot of different sugar groups, including those gangliosides that I was talking about a few slides ago. And so that prompted us to make vesicles out of cell membranes, so these GPMVs. And I know all of you are familiar at Michigan are familiar with this because you have an expert in your department there in Sarah Veach in terms of making and characterizing these giant plasma membrane vesicles. So we made these, and the reason they're really nice as a biophysical tool is because they form these large vesicles that are optically resolvable. So we have a nice platform for looking at interactions of proteins with cell membranes, but it's simpler than a cell in that we don't have to worry about all the active processes that are occurring in a cell that might alter how our protein is interacting with the membrane itself. So we first started by taking fluorescently labeled alpha-synuclein. Oh, my pop-ups are a little bit out of order. So focus on this this one up here now. So we started off by making these vesicles out of SHSY5Y cells. So these are a neuroblastoma line that are commonly used for studies of Parkinson's disease because they recapitulate a lot of the same pathways that are active in Parkinson's disease. And what you can see is as we add our fluorescently labeled synuclein to these cells, to these vesicles, not only do we get binding to the vesicles, but that as we add more and more protein, we start to get sort of clustering of the vesicles and also these clusters. So it's not binding uniformly along the surface of the vesicles. And Melissa was able to quantify this. And the way she did this is we would take a sample chamber and she would park her focused laser somewhere in the media, and then we would add our alpha-synuclein, and she would measure the loss of sample in the media. And so we could use that to ascertain how much of it had bound to the vesicle. And so that's what's seen over here. This 80 is the number of molecules we'd expect to have in our focal volume if none of it was binding. So after about 5 minutes of incubation, she was down to 20. So a lot of it was bound. And I just want to contrast that to what happens if we make our GPMBs out of HEC cells. So these are the human embryonic kidney cells. You can see the only reason we can actually see these vesicles is because there's so much fluorescent alpha-synuclein not binding. So we see it in the background, right? And then we were, I'm going to tell you, we were barking up the wrong tree when we were looking at the gangliosides, but it did have us asking, what is the component of this SHSY5Y membrane that is driving this specific interaction between alpha-synuclein and the SHSY5Y cells as opposed to the HEC cell GPMBs. And so there's clearly going to be different proteins expressed in HEC cells or in SHSY5Y cells. And so we kind of focused on the sugars to begin with because of this interaction with BOG. So what we did is we treated these GPMBs with various different endoglycosidases. And I will say we tried 4 different ones. We tried 2 of the heparinases, which cleaves the heparin sugars. And then we also tried a mannose cleaving sugar. And then we also tried P and GSF. So P and GSF, of course, is the one that turned out to be most interesting, which is why I'm showing you data for it. So if you want to contrast the alpha-synuclein binding to these P and GSF treated vesicles compared to those that were not treated, and the quantification over here. So the fact that we have more synuclein left in solution means that we have less of it bound, but you can even see that by eye, right? We don't see any clustering of the vesicles. We see weaker binding. We see that it's more uniformly bound to these vesicles. And P and GSF is an enzyme that specifically cleaves complex end-linked glycans. So hopefully you can see this arrow here. So end-linked glycans, of course, modify asparaginase. And it cleaves before the first glycosidic bond of these complex end-linked glycans specifically. So this structure I'm showing you here is the common structure of these end-linked glycans, and they get modified by all sorts of side groups up here. But this is the common structure. This fucose group is not always there, but can be there, but the enzyme is independent of whether or not that fucose is there. So the effect we saw was really specific to the treatment with the P and GSF, not the other endoglycosidases. And I'm going to point out that there have been a number of studies which have implicated heparin and sulfates. So these GAGs as being important for cellular interactions of both alpha-synuclein as well as other amyloid fibers such as tau. But in our hands, at least, the end-linked glycans seem to have a much more specific interaction, and that's what I'm going to tell you more about. Okay, so we, having seen this interaction in the GPMBs, we were interested whether or not it was also found in live cells. So now I'm showing you images of these live SHSY5Y cells where we've added fluorescent alpha-synuclein. And in the top panel, this is where we have not treated the cells with P and GSF. On this left-hand side, I'm showing you monomer alpha-synuclein. And on the right-hand side, I'm showing you these PFF, which is just preformed fibers, so fibrillar alpha-synuclein. And what you can see in these images is that when we've incubated for 12 hours with our fluorescently labeled alpha-synuclein, either monomer or fiber, we see these inside the cells, we see these bright puncta, right? So this is somewhere after having been endocytosed, it's somewhere in the endocytotic pathway. You can see that it's still in these little vesicles after having been internalized. But strikingly, if we treat the cells with PNGASF, what you can see is that we no longer see internalization of the protein. I will say we did check to make sure that we weren't otherwise disrupting endocytosis. If we add transfer into these cells, for example, it gets in just fine. And so that's sort of the canonical way of measuring that catheter-mediated endocytosis is still functional in these cells. So we hadn't otherwise disrupted endocytosis. We had just stopped uptake of both monomer and fibrillar alpha-synuclein. Melissa was able to quantify this a number of ways, but one of the ways she did this was really similar to what we did with the GPMVs, where she parked a focused laser in the cell media outside the cells and added protein and then watched the loss of protein. So we get some kinetic information that way. And so what you'll see is that, after about 5 hours in the untreated cells, we've internalized, most of the protein, whereas this process becomes much, much slower. So this is an important point, right? Like this is the 12-hour point, which is what I'm showing you here. We haven't completely eliminated uptake, but we have certainly slowed it down. So that does suggest that there may be primary and secondary pathways that are important here. Okay, and consistent with what we saw with the GPMVs, if we incubate alpha-synuclein with HEK cells, we don't see any internalization. And I would say this is broadly consistent with literature. In other cases where people are using HEK cells as model systems for fiber or monomer uptake, a transfection agent is typically used in order to introduce the protein. So what we're just to make sure that's clear, we're looking at spontaneous internalization. We're not using a transfection reagent. We're just adding the protein to the cells, and we're quantifying the effect of internalization. So it doesn't bind to GPMVs made from HEK cells, and it also doesn't seem to be internalized by HEK cells. We verified this in neurons. So these are primary hippocampal neurons from mouse. You can see the images look a little bit different. So as it turns out, when synuclein is internalized by neurons, it actually escapes from those endocytotic vesicles a lot faster than it does from SHSY5Y cells, which is kind of interesting on its own. So what you see instead is sort of a diffuse fluorescence in the cell body and processes, as opposed to those puncta we saw previously. So we had to quantify a little bit different, but you can see that the results are largely the same, right? If we have not treated with PNGASF in this top row, you can see that we see bright fluorescence for both the monomer and the fibrillar protein. And if we treat with PNGASF, we dramatically reduce the amount of protein that gets internalized. And that's just quantified over here. And then lastly, we were interested in whether or not these differences in internalization had any sort of impact in terms of propagation. So just to take a step back and ask, why would we care so much about alpha-synuclein being added externally to cells? And that's a really good question. And I think there's two primary reasons. So one is that it appears that alpha-synuclein, although it's generally described as a synaptic protein, seems to be outside of cells for at least some part of its lifetime. And what I mean by that is in cell models, the detection alpha-synuclein is generally detected in the extracellular media. And in fact, it's been shown that in iPS cells, for example, iPS lines that express multiple copies of the synuclein gene, so triplication of the gene, which is also seen in disease, compared to just the normal 2 copies, has more synuclein extracellularly than the cells expressing the regular level of synuclein. So it just suggests there's some sort of concentration dependence. The other reason is that in the amyloid field, I'm sure as many of you know, there's a general idea that propagation of pathological material may occur through release of toxic species, whatever forms they may be, fibers or oligomers, into extracellular media, and then these get internalized by the surrounding healthy cells, which then causes aggregation of the soluble protein inside those cells. So there's both from the sort of normal function, the fact that synuclein seems to be found extracellularly at least some part of its life, as well as the fact that this may be relevant to disease propagation, are both motivations for us looking at extracellular alpha-synuclein. And so what I'm showing you now are actually, there are antibody staining of fixed neurons. This is our control up here. And so what we're doing is staining these neurons with this phosphoserine 129 antibody. So this is an antibody that is typically used to detect pathological aggregates, because serine 129 is the phosphorylation is upregulated in aggregation. And then this beta tubulin is just allowing you to see that our cell structures still exist. So this is day 7 normally of our cells, of our neurons, where you see very little of the phosphoserine 129. If we have treated those cells with acetylated protein that has PFF, so our preformed fibers, you can see by day 3, we have a lot of the red staining in the cell body. And then by day 7, it's basically spread throughout the processes as well. And it's actually quite intense. And we couldn't measure beyond day 7 because by day 8, most of our neurons were dead. And this is just a contrast with what happens if we treat these these cells with unmodified protein. So this is protein lacking the acetyl group, but otherwise identical to the PFFs that we're looking at here. And what you'll see is really striking. That day 3 of treatment looks more or less like control. And it's not until day 7 that we begin to see increased staining of phosphoserine 129 in the cell body. And we could continue to measure these up through at least day 10. So the pathological, I guess, implications here are that the acetylated fibers are much better at being able to induce aggregation of the endogenous protein. And that doesn't mean that it's a better seed necessarily. It could simply be a matter of kinetics, right? If this protein is getting into the neurons more rapidly than the unmodified protein, then it just basically starts at an earlier time point in its ability to seed aggregation. So this showed us that clearly there's a strong difference in how the acetylated fibers act compared to the unmodified fibers. This is actually a fairly important point because a lot of the biophysics and biochemistry that's done on alpha-synuclein doesn't use the acetylated protein. And we can see very clearly here that there's a major difference in how it impacts the sort of at least the timing of the pathological features of uptake of fibers. And what was even more striking is we went back and revisited all of the experiments that we'd done with the acetylated protein. We found that all the effects that we saw were really specific to acetylation. So I'm just going to go through an overview. So if we add the unmodified alpha-synuclein to the SHSY5Y vesicles, it binds more weakly, and its binding is not impacted by treatment with PNGSF. And this is just a reminder of what under the same conditions using the acetylated protein look like. So we quantified that, and it certainly binds much less. This is actually data I didn't show you previously for the acetylated protein, but this is cleaving the glycans off of the cells and then adding the purified glycans into the media and using FCS to look at binding. And you can see the increase in diffusion time of the acetylated protein and no effect of the unmodified protein as we increase our glycans. If we add the unmodified synuclein to cells, it certainly gets internalized, both monomer and fiber, but it doesn't change when we treat it with PNGSF. So you can see internalization of both types of protein, regardless of whether or not we've treated it. And this quantification is shown on the same plot we showed you earlier, where this gray, I'm sorry, I'm missing my figure legend there. The gray is the acetylated protein, and the orange is the acetylated protein following PNGSF treatment of the cells. And the red and the black are unmodified protein plus and minus PNGSF treatment. And you can see there's essentially no difference in the internalization, so it's completely unimpacted. You'll also notice that for the untreated cells that the internalization of the unmodified protein is slower. And that's sort of our basis for the difference and the effect of, in this previous slide, here was that the acetylated protein seems to get into cells much easier than the unmodified protein. And then lastly, this is also true in neurons. So you can see that in the absence of PNGSF treatment or the presence of PNGSF treatment, we basically have the same extent of internalization of this protein. It really doesn't depend on the presence or the absence of the glycan. So this is pretty striking for two reasons, right? So just to summarize what we've shown you so far, is that the uptake of the spontaneous internalization of alpha-synuclein by neurons or neuron type cells, such as the SHSY5Y cells, is dependent not only upon the internal acetyl group of alpha-synuclein, but also the inland glycans that are found on the surface of the SHSY5Y cells or the neurons, right? So this basically suggests that there is a specific glycoprotein or glycoproteins that synuclein is binding to or interacting with that's important for its internalization. So this is not a new idea. There's been a lot of really nice work which is looking at identifying possible receptors for amyloid fibers. in different cell types that might be involved in their internalization. I will say most of the studies have focused on the fiber as opposed to the monomer. So one thing that's different about our work is that we see, at least from the level of looking at the overall glycome of the cells, that the unmodified protein, the monomer protein and the fibrillar protein behave similarly. But it did have us asking, what could the possible receptor for acetylated, or what acetylated synuclein be? And that led us to the identification of this protein called Neuroxin. And I'll actually say it wasn't us that identified Neuroxin. There was a paper from the Dawson group a couple years ago where they identified Neuroxin as well as LAG3 as specifically driving uptake of alpha-synuclein fibers. But the majority of their work focused on the LAG3 protein. They didn't really look at Neuroxin beyond identifying it. So we were screening some of the proteins that had been previously identified by other groups as deriving uptake of alpha-synuclein, and that's how we ended up at Neuroxin. So up in the corner here, I'm just showing you the hex cells again, and this is important because it's just a reminder that hex cells do not spontaneously internalize alpha-synuclein. So that makes them a really nice platform, because then we can selectively express proteins of interest in these hex cells and then determine their impact on alpha-synuclein uptake, which is exactly what we did. And that's what I'm showing you with Neuroxin here. So there's a lot of data, so I'll just go through it sort of row by row. So we're starting with the acetylated protein in the top two rows, monomer and then fiber. And then the unmodified protein in the bottom two rows, monomer and fiber. All of the cells are expressing Neuroxin, which is labeled with GFP on its intracellular terminus. And then the alpha-synuclein is labeled in red. And so what you can see really clearly, I hope, from these slides is that when we have Neuroxin expressed in these hex cells, that the acetylated protein is internalized. You see lots of bright puncta inside the cells. Whereas for the unmodified protein, we really don't see any internalization. And the other thing you can see this really nicely is if you look here at the cell membrane here, this is where Neuroxin should be, right? It's a plasma membrane protein. It's actually typically found in the presynapse in neurons, but when we express it in hex cells, it goes all over the plasma membrane. And you can see that in this case where there's no internalized synuclein, it's all on the cell membrane where you'd expect it to be. Whereas in these cases where synuclein is being internalized, it's also driving the internalization of the Neuroxin, right? So we see overlays inside these puncta of both Neuroxin and alpha-synuclein. So it's driving it off the cell surface. You still see some of it there, but you see a lot of it inside these bright puncta inside the hex cells. And then very strikingly, when we treat these hex cells with PNGase-F, not only is our Neuroxin now back on the membrane where it should be, we see very little, if any, internalization of alpha-synuclein. And so we've identified this Neuroxin as being capable of driving uptake of alpha-synuclein in what I would call a chemically specific manner, meaning that there is some specific interaction that is driven by the acetylation of the alpha-synuclein and the glycan group on Neuroxin. And there is a glycan group on Neuroxin. In fact, there's a single N-link glycan on Neuroxin on its extracellular domain. And I'm going to tell you a little bit more about that in a minute or two. All right. Okay, so this is sort of the summary of where we are in terms of really our understanding of the story. And I'm going to tell you just a little bit more about this last point. But these are, I would say, rather than having sort of conclusions about what we've done, we've just generated a lot more questions. So the first thing is that it appears that alpha-synuclein, when it's acetylated, is a carbohydrate binding protein. And so that's a unique function for alpha-synuclein that hadn't been identified before. As I've already motivated, I hope that this interaction with extracellular glycans and glycan proteins may be important for alpha-synuclein's function. If it's found extracellularly at some point in its life cycle, then it may need to be re-internalized. And it seems that the acetyl group is important for that recognition. And then for fibers or disease mutants, it may be relevant to sort of propagation and disease. We're working on high resolution structural approaches to understanding this, this interaction, because we think it's really interesting to understand how this acetylation is going to confer this glycan specific binding. There's been nice NMR work from a number of groups which have shown that acetylation of this N-terminus of alpha-synuclein enhances the helical content, although it doesn't form stable helices in solution, it is more helical, and this may be important for recognition, and we're looking into that. And then this last is there any kind of physiological significance between the interaction of Neuroxin 1 beta and alpha-synuclein? And that's what I want to tell you a little bit more about, because that's something we're super excited about. So what is Neuroxin 1 beta? Neuroxin 1 beta is a presynaptic protein. So just to sort of orient everybody quickly, we have, this is just the cartoons of neurons where the axons are making connections with the cell body. And these blow up of this connection is going to look something like this, where you have the presynaptic side and the postsynaptic side on the different cells. And the connections between this presynapse and this postsynapse are maintained by the synaptic adhesion molecules. Neuroxin 1 beta is one of these presynaptic adhesion molecules. It's shown here in a little bit purple here. It has a number of different binding partners on the postsynaptic side. One of the primary binding partners is a protein called neuroligin, which is shown here in orange. And so maintenance of these connections is really important for maintaining synaptic stability. And then, of course, a changing of these connections is going to have a role in synaptic plasticity. And this is part of why we got so excited, given that synuclein's initial function had been identified as having a role in learning of bird songs. And the idea was that it was somehow modulating synaptic plasticity in those birds. And so fortunately, there are crystal structures of the major extracellular domains, both of beta Neuroxin and of Neuroxin 1 beta and neuroligin, and that's what I'm showing you here. So this is their binding interface here. Neuroxin 1 beta has a single globular extracellular domain. There are several different splice isoforms, which I'm not going to talk about. The glycosylation is on the single extracellular domain. Neuroligin also has a single neuroligin 1 has a single extracellular domain. It's also modified by glycosylation, although we haven't seen that to be too important for the interaction. So these will bind across the synaptic cleft. And so that led us to question whether or not synuclein could modulate the interactions between these two proteins. So first of all, something quite interesting is that synuclein actually will bind to neuroligin. So this is hex cells expressing neuroligin. This top row is in time 1 hour of incubation. This is at 6 hours of incubation, and they'll become important in a minute. And what you can see is that the alpha-synuclein is bound on the outside of the plasma membrane when we overexpress neuroligin, but this doesn't really change as a function of time. The images at 1 hour look more or less like the images of 6 hours. And this is just to remind you, this is in contrast to what we would see with Neuroxin, right? At Neuroxin at 1 hour, we mostly see it on the cell surface, but by 6 hours, we see a lot of the proteins beginning to be internalized. We see a lot of these puncta inside. So although synuclein binds to neuroligin, it doesn't seem to drive uptake. And I will say this is not a general feature. We've expressed a lot of different proteins in hex cells, and the interaction between the monomer protein and neuroligin is not, I'm sorry, we don't typically see binding. So it's not that alpha-synuclein binds to anything you express in a hex cell. So this does seem to be a unique interaction for neuroligin. It's just different from the one for Neuroxin. But most interestingly, it seems that alpha-synuclein is able to interfere with the Neuroxin and neuroligin interaction. So this is not beautiful images. I know that this is preliminary data, so this is unpublished data. I just wanted to share it because it's exciting. These are hex cells that we are expressing either Neuroxin 1 beta in, and these are hex cells who are expressing neuroligin. The reason they look like they're dead is because for these assays, you basically have to cleave them off of their adherent cells, and you have to cleave them off the plates to get them into solution. And so they're not rounded up because they're dead, they're rounded up because we've just cleaved them off the plates. And what you're looking at is the cells that have settled back down on the surface of the plate, but they haven't spread out like they normally would if they'd been sitting there for a while. So the reason we do it this way is because we can then mix these Neuroxin 1 beta cells and these neuroligin cells together. And if we do that in the absence of alpha-synuclein, you can see big fat clusters of cells that settle down on the surface. And the reason for this is that it's driven by the interaction between the Neuroxin 1 beta and the Neuroxin 1 alpha. The density of cells in this experiment are the same as the density of the cells here. And the reason we see so many more cells here is because they're bigger clumps and they settle down a whole lot faster. And what's really striking is that if we treat this with the acylated alpha-synuclein, we basically get disruption of these clusters. So you can see with 10 micromolar protein, we already have a fairly significant reduction. And with 20 micromolar protein, the clustering is reduced even further. And consistent with everything else that we've seen so far, this seems to be really specific to the acetylated protein, right? And so that the unmodified protein, which I'm showing you here, at 20 micromolar doesn't seem to have any effect. At 100 micromolar, maybe we're starting to get some disruption, but it's certainly not as striking as what we see at much lower concentrations of alpha-synuclein. So this is interesting to us for two reasons, and it goes back to both dysfunction and function, as I've sort of been talking about in parallel this whole time. One is that if alpha-synuclein has a functional interaction with these proteins as part of its native function, then it may have some role in help regulating their interactions, and that this is a potential point for dysregulation in disease, meaning that if normal alpha-synuclein levels are meant to regulate this interaction, then an increase in alpha-synuclein or the presence of alpha-synuclein fibers may be able to dysregulate the interaction. And if you think about, I think what's another sort of striking thing about our work is that the fiber and the monomer have largely behaved the same in all of the assays that we've done. And this is surprising on the surface, but when you think more about the parts of the protein that may be important for this interaction, although we're still working to identify those specifically. In most fiber structures, the N-terminus of alpha-synuclein is unstructured and accessible, and the N-terminus is where the acetyl group is. So what we're hypothesizing is that the primary domain of alpha-synuclein that binds to and recognizes Neuroxin 1 beta is that N-terminus, and that acetylated N-terminus specifically. And this should be present both in the fiber and the monomer. It's just the fiber presents a really dense multivalent set of those those alpha-synuclein binding sites, and therefore the overall impact of a fiber may be much stronger than that of a monomer because it presents a high density of alpha-synuclein binding sites. So that's hypothetical. We're still working to figure that out specifically, but I think it's less surprising to consider that you see the same effects for monomer and fiber when you think about the regions of the protein that might be involved. All right, I'm going to end it there. I am happy to take any questions, and I will turn my video back on so that you can see me at least. All right, thank you.
Thank you very much, Liz, for the great talk. So there are two ways to ask questions. You can raise your hand. We can enable you to ask the question directly to the speaker, or you can type in the question in the Q&A folder. One of us can ask question to the speaker. So Liz, let me get started on this. Great talk, really enjoyed it very much. It looks like lipids per se do not have a big role on Neuroxin synuclein interaction and uptake of synuclein inside the cell. And what about the role of synuclein oligomers? Are there any?
Yeah, so we haven't tested oligomers, and I would say even the fibers are harder to interpret, right? So our expectation is that when you add fibers to cells, that it's primarily the smaller assemblies that get internalized, not the really big fibers, right? So this is, we follow this protocol that was established by Virginia Lee, where you aggregate your sample and then you sonicate it and then add the sonicated fibers. She showed, and a number of other people showed, that the protein goes in much better when it's sonicated. Our interpretation of that is that smaller things get in better. And so we don't really know what's getting in what species get in. We've done TEM of our fibers after sonication, right? And you see a mixture of big and small things. We don't know which of those are the ones that get internalized. And so some of those are potentially closer to oligomers than fibers, but we haven't characterized that. And I don't honestly know a really great way to characterize that.
So do you expect Neuroxin itself to oligomerize or fibrilize?
So that's a great question. I mean, clearly in the GPMVs, we saw these big clusters. And so those were done, and we saw that, and I again did show data for this. If you make GPMVs out of hex cells expressing Neuroxin, then you also see that. But what you're missing there is the cytoskeleton, right? Which would normally provide some structure to the proteins that are found in the plasma membrane. And so it certainly can cluster Neuroxin in the absence of a cytoskeleton. I don't know how effective it is at clustering it in the presence of a cytoskeleton. And Neuroxin itself, at least binds to neuroligin, I think, as a monomer. I think neuroligin dimerizes and Neuroxin is a monomer. So at least for that binding interaction, it's not oligomerizing, although we can't rule it out as being important for uptake of alpha-synuclein.
Great, thank you. So there's a question from Long Cheng. You can ask the question directly.
Oh, can you hear me?
Yep.
Yes.
Yeah, so great to talk, very exciting. So I have a couple questions. First question, in your model, actually, the cell intake, the alpha-synuclein, no matter it's a monomer or the PHF is, I think, is a free, no vesicle coated. And think about the propagation. And if you have aggregation form, then had to secrete out the cell and to intake by another neuron. So my question, do you think for the propagation, what form of the alpha-synuclein will be, whether they're free or in actual cellular vesicle, and whether they will be for the same intake maximum or different one? That's the first question. The second,
let me answer that before you go on, just because that's a lot of question, and it's a super question, and I would say we have very little.
okay.
We have very little, we don't know. Like if you read the literature, we haven't looked at this directly ourselves, but a lot of people have looked into this, and you're right. People suggest that synuclein gets secreted in extracellular vesicles. It certainly doesn't, it's not secreted by canonical secretion pathways, like it's not a canonically secreted proteins, but there seems to be a growing number of these sort of non-canonical secretion pathways whereby proteins like alpha-synuclein are released into the extracellular space, and whether or not they're encased in some sort of lipid membrane or on their own, I think, depending which paper you read, you'll get a different picture of this, right? So we don't have a clear understanding of it, although certainly we have done some work in neuron models where we're just detecting extracellular alpha-synuclein, and we know that we can see monomer out there, but that doesn't tell us it's the only, like monomer not in a vesicle, but that doesn't tell us that's the only form by which it gets out. And you can imagine when cellular stress pathways get activated, you're likely to change the normal secretion anyway. So that's a complicated, great question. I wish we knew about it.
And the other things for intake alpha-synuclein basically happen to two neuron type. One is neuron, one is microglia cell. So my question, what happened to microglia cell?
Oh yeah, so these are treated with chemicals to suppress microglial growth, and the reason we do this is because the glia gobble up everything, right? So if you had glia growing there and you added any protein, it would get eaten up by the glia so fast you would never be able to see it on the neurons. But that's actually, I think there's a growing interest in any role glia may have in general neurodegeneration, and so that's also probably something worth investigating.
Yeah, so last quick question. Do you think this Neuroxin and is in disease model is loss function or gain of function? And the loss function will be like a dysfunctional synapse. Gain of function will be you have more intake and more propagation.
Yeah, so we have, right, so that there's a couple of different ideas we have. If we would like to believe that maybe Neuroxin and alpha-synuclein interact functionally, then, there's sort of two, one is that synuclein could bind to it and physically block its ability to interact with other proteins. That's what we think is happening in the neuroligand assays, because those are after incubation of only half an hour, which is not long enough for alpha-synuclein to have been internalized, and therefore we're not seeing loss of Neuroxin 1 beta from the cell surface on that time scale. But then that's the other possibility. If alpha-synuclein binding drives Neuroxin uptake, even though we think Neuroxin will normally get recycled back to the cell surface, that's what normally happens to Neuroxin, that if this is overwhelmed, such as in the case of an increase in alpha-synuclein concentration, or let's say the fibers are much better at driving more Neuroxin off the surface, then you might also have loss of function due to internalization of Neuroxin and not returning to the cell membrane in a timely manner. But that's us waving our hands around. We're investigating that. We don't really know.
Thank you so much. It's a great.
Glad you can ask the next question.
Yeah, Liz, that was a very impressive talk. I was particularly impressed by how far you dive into cell biology now. And my question is about those puncta that you see inside the cells when you have Neuroxin and alpha-synuclein internalized together, the acetylated version. And do you know, or do you have any idea whether these are still inside the endocytic vesicles or whether they go back into the cytoplasm?
So the cool thing is, so in neurons, they clearly go back in the cytoplasm, or at least that's the only sort of viable mechanism we can think of whereby they have to get out of the endosomes if they're going to cause seeding of the endogenous protein to aggregate, right? And SHSY5Y cells, it could just be that it's a lot slower and they eventually escape, or it could be that the pathway is different and it's going to get trafficked to the lysosome and get degraded. I will tell you that we've done something cool that Melissa did right before she left, is she did longer time points in those studies. And you see after about 24 hours that the Neuroxin and the alpha-synuclein are no longer co-localized, right? So they begin to diverge in terms of their pathways. So we think the Neuroxin is probably getting recycled back to the cell surface. We don't know what's happening synuclein, like if it's going to be able to escape then from the endosomes or if it's getting trafficked off to the lysosome. So they do diverge, but we don't know what happens in the model systems. We're assuming in neurons they're escaping because you see them all throughout the cell body.
Okay, thank you.
Okay, Bin, can you ask a question? And following that, Dave will ask a question.
Sure, a very interesting talk, Liz. Congratulations. So I have a question regarding the acetylation to the aggregation of synuclein itself. So does the N-terminal acetylation actually promote aggregation of synuclein? Maybe say change the biophysical property of the aggregation of synuclein itself?
In vitro, we haven't done this, but other people have. In vitro, it actually slows aggregation a little bit. So if you're talking about de novo aggregation of alpha-synuclein, the acetylated protein aggregates a little bit more slowly than the unmodified protein. So that's why we attribute the enhanced seeding to the enhanced uptake. The protein still gets into cells better, and therefore it's capable of seeding aggregation more rapidly than the unmodified protein, but that's probably not because it is more inherently aggregation prone than the unmodified protein.
Does the second question is the acetylation, because there are other lysine in the synuclein sequence and N-terminus, C-terminus, do they have any a pathological role in the disease?
So our motivation for the N-terminal acetylated is that we would argue that it is the physiological form of the protein and therefore the one that should be studied, right? If this modification occurs co-translationally and is ubiquitous, and that's been shown by mass spec studies. So, all of these other modifications are typically found in relatively low abundance by mass spec, but the acetylation is always there, right? So it's the dominant one. We'd argue it's physiological. There is acetylation of side chains. There's some relatively recent work from Hilal Lashuel where he has looked specifically at, and they're lysines close to the N-terminus, although right now I'm not remembering exactly which ones. And those I think are thought to be, regulated acetylations, right? So you have an increased abundance of those acetylations in disease. But that's sort of in contrast to what happens with the N-terminus.
Thank you.
Mm-hmm.
Okay, Dave, and then Wang. Dave, go ahead.
Liz, for reasons your postdoc advisor pointed out, I understood very little of your talk. But
there's single molecule fluorescence in this one.
yeah, or anything that I could sort of hang my hat on and think about. But just to get some point of information, how many acetylation sites are there in the C-terminus of alpha-synuclein?
So the N-terminus, it's the N-terminal methionine that gets acetylated on the that N-terminal acetyl group. The N-terminal group, and the lysine side chains in the N-terminus also get acetylated. I think it's lysine 5, and I honestly I don't remember. There's several in the N-terminus that get acetylated, but not constitutively. So those are regulated by, and I think part of what Hilal was looking at was the interaction of some of the deacetylases in terms of removing those modifications on those lysine side chains.
I assume there are ATP-dependent enzymes that are actually doing this acetylation. Is that correct or not correct?
Yes, so the enzyme is very specific for the N-terminal sequence. So for example, this NATB, which is the one that acetylates alpha-synuclein, is specific to the methionine, and then it has some sensitivity to the flanking residues of that methionine. So it won't acetylate things that don't have sort of the right flanking residues. So there's a whole host, there's at least through NAT-E, maybe more than that, NAT-F, and they all have different sequence specificity in terms of what they will, and those are just N-terminal acetylases, and in terms of what they recognize and will acetylate.
And do you know, or does anyone know the mechanism of those acetylation timescales that are involved, and things like, I don't know how correlated those enzymes are?
Oh, so I mean that's well beyond what I know about N-terminal acetylases. I will say what happens in our system is probably different than what happens under when it's being expressed under in a normal mammalian system, because we're doing co-expression of a yeast NAT-B in E. coli. And so it's unlikely that yeast NAT-B actually recognizes the E. coli ribosome and associates with it. So we're kind of assuming, although physiologically acetylation should occur co-translationally in this E. coli system, it's probably occurring post-translationally because the enzymes are not going to associate with each other, which means that the mode of recognition, there's a lot of open questions there. I mean, the through collaborators of ours just solved published a structure of the N-terminus of NAT-B bound to the N-terminus of alpha-synuclein. They were able to trap it by covalently linking acetyl-CoA to the N-terminus. So you could see it, but what it tells you is kind of what we typically know about alpha-synuclein. It doesn't really become structured. You can see maybe the first 10 residues that are associated with the NAT-B and then nothing. So that's all. I'm not. That's beyond what my biology understanding of NAT-B. Otherwise.
All right. Thanks.
Thanks, Dave. Wang, you are next.
Me? Okay. Yeah.
Go ahead. Yeah.
I have several questions, which most of them are already asked. So let me just ask one about the N-glycans. So do you think that has a similar mechanism as tau with heparan sulfate, which need a sulfation and acidic charge? Do you know if the acidic sugars like sialic acid is involved in this, interaction?
Right. So I would say, with the tau and the heparan sulfate, right? I think that is specific for tau fibers, right? Not tau monomer. And the difference between the glycosaminoglycans and the N-linked glycan is that those are long repeat chains. And you're right, there's been some, I guess, specificity in the terms and the types of GAGs that are best at driving uptake. But in our hands, we think there is probably a specific subset of glycans that it interacts with, and it's a specific interaction because we don't see any kind of binding in the absence of N-linked glycans. So I guess my view of the glycosaminoglycans is that those are generally very negatively charged polymers, and that you'll have a lot of positively charged things bind to them. And so it's harder to sort of parse out exactly what that interaction looks like. So yeah, I don't have a great answer for that. And we think that probably GAGs are involved in a secondary way for uptake of fibers for synuclein, although not in the primary way that the N-linked glycan seems to be. But we haven't parsed all that out yet.
Can you try to block the interaction by lectins? Like if you use WGA, which will bind to sialic acid terminal, sugars?
Oh yeah, so we haven't done that. That's a great idea. I would say the sort of the result that I haven't shown you because we have not been able to reproduce it, but was also something that we got a long time ago, we actually sent this off for a glycan screen. So the Center for Functional Glycomics at Harvard ran a screen for us where they have 600 glycans dotted on a chip, and we sent them fluorescent protein, and they sent us back the most amazing results, which was they'd identified 6 glycans, which the acetylated protein bound to, and the unmodified protein didn't bind at all. And none of those had sialic acid on them, which is why we haven't pursued sialic acid, although it would we probably ought to look more into it because they have not been able to reproduce those results. And so we don't, maybe it was a fluke, we don't know. But that's sort of what turned our focus away from the sialic acid, and we haven't gone back to it, but that's a great point.
I mean, you can easily buy those and, lectins. And do you think this getting in and out of alpha-synuclein happens only in synapses? Because the Neuroxin 1 is presynaptic protein.
Yeah, so we do think that. So we have some more recent work on neurons, incubation with neurons, and looking at co-localization with Neuroxin after staining. And we certainly see in those studies that even the monomer protein, so this is not fibers, it co-localizes in puncta with the Neuroxin after it's internalized. And you can actually see it being trafficked down the processes, which suggests to us that it is only occurring at the synaptic termini. But that's really preliminary. Yeah, so and just to be clear too, you know, while we think that Neuroxin may be a primary specific binder, it certainly doesn't rule out other pathways that may especially become dominant under conditions of disease.
Thank you.
Thanks, Juan. Liz, can we take one more question and then we'll
Sure,
move
yeah,
on
sure,
to
that's fine.
the informal session? Okay, Vijay, go ahead, ask a question.
Hi, Liz. Good to see you. And
Okay,
wonderful,
BJ, yeah.
hi, wonderful talk. I have a question, a couple of questions. First is you might have answered this before, and the N-terminal acetylation, how much of that aggregation state of alpha-synuclein is affected by that? And then concomitantly, how does that affect internalization of alpha-synuclein?
Right. So in terms of, I mean, other people have solved like structures of the acetylated fibers, right? So there have been at least 2 or 3, if not probably more of those actually, now that I say that. Right. And so in those structures, the N-terminus is, you know, the structure part of the protein doesn't start before residue 45 or 50 or something. So the N-terminus is still disordered and accessible. In terms of kinetics of aggregation in vitro, the acetylated protein is a bit slower than the unmodified protein. And so we can't make a direct sort of correlation between or draw from that in vitro to the cell uptake, because when we add the fibers to the cells, they're already aggregated, right? And we know from TEM that the aggregates are, you know, after sonication are roughly the same size and distribution of sizes. So, you know, within some crude estimate, we're adding the same material, both for the unmodified and the acetylated protein, and the acetylated just gets internalized much more rapidly. And we would argue that that's due to the specific interaction with Neuroxin, but you know, there's some more work to be done there.
Great. The other question is just curious, have you looked into the advanced glycation end products as a process by which they get internalized? It is known that these age products coming from sugars are known to sort of assist internalization of alpha-synuclein.
No, that's a really good question. Oh, I guess I thought what you were going with that was like, you know, changes in the glycosylation patterns of cells as a function of disease. So that's something that's been suggested, at least for Alzheimer's, that you do get changes to those N-link glycans, which again could modify this interaction. We haven't done anything with that, although I think that's a really interesting idea.
Thanks, great work.
Thanks.
Okay, there's a lot of questions, but let's formally thank Liz for a great talk and thank the panelists, Ari and Duncan and Gilad, for making this talk more beautiful and more special. Thanks for all the participants, and hope to see you then on next seminar, next ZOOMinar next Saturday. So let's go to the informal session. People can join us in the panel. There's Bina waiting in the line to join. Bina, you can go ahead and ask the question directly to the speaker. Bina, go ahead. Hello, Bina. Ari, do you have a question?
Hi. Can you hear me?
Go ahead. Yeah.
Yes. So I'm just curious that when you add PNGS and the sugar is gone, you don't see the internalization, right?
That's right.
But
yes.
there must be so many proteins, membrane proteins that carry the sugar. Then how come where is the specificity coming from? That would make it that Neuroxin should play a role in internalizing the aggregates, right?
Yeah, that's a great point, right? So the BNJSF is like hitting it with a hammer, right? We're cleaving off all of the glycans there. And we know that we're getting most of them because if we then stain with concanavalin, which sort of, you know, generically binds more simple sugars, you see a huge drop in the binding of concanavalin, right? So it's not a direct path from seeing that the N-link glycans are important to Neuroxin. We did we basically screened a number of different proteins to see whether or not they showed any glycan specificity. And actually, I will show this is just to maybe also to help understand further emphasize the specificity of our interaction. I mentioned this LAG3 as one of the proteins identified by the Dawson group, and that was what they focused their paper on. So we were interested to see whether or not LAG3, we saw the similar effect with LAG3. So here we're expressing LAG3 in our hex cells. And this is again monomer acetylated, fiber acetylated, monomer unmodified, and fiber unmodified. And so in the Dawson screen, they were specifically selecting for proteins that internalized fiber over monomer, although they were not using acetylated protein. But what you'll see is that we basically recapitulate their findings, and that we see uptake of acetylated fiber, we see uptake of unmodified fiber, but we don't see uptake of the monomer protein in either case, right? And then if we treat this with PNGASF, it basically has no effect on uptake, right? So the fact that the removal of the glycan on Neuroxin is, or we're interpreting it as a removal of Neuroxin, is specific to the acetylated protein is bolstered, I think, by this case where we find another protein, and we can only do this for the fiber since the monomer doesn't get internalized, but it really doesn't have any impact on uptake of fiber. Does that, I mean, so I agree with you. We're still, the thing we're working on now is actually, we're making stable hex cell lines with a version of Neuroxin where we've mutated that asparagine so that it can't be glycosylated. And we're going to determine whether or not that, you know, that will be really a much cleaner result in terms of showing that it's specifically the Neuroxin glycan as opposed to other glycans that are important.
Could I ask one more?
Go ahead, go ahead.
So I think it's also interesting if it, like you mentioned earlier on, that maybe, you know, about 70 to 80% of proteins seem to be N-acetylated. So could this be pointing to a novel or a different mechanism of protein intake into the cell, that, you know, the acetylated proteins could probably now can, you know, enter into a cell using this sugar acetyl? I don't know. It's not really proven that it's the acetyl group interacting. I'm not sure. So have you thought along this line to see that this could be some sort of another generic mechanism for uptaking proteins?
That's a really, really great question. And I'll tell you what we have done. So we think the effects first, I mean, because synuclein is disordered, we think the effect is, we think this enhanced helicity conferred by the acetylation is important. And the reason we think that is we've made a variation of alpha-synuclein where we replace that, the N-terminal methionine with an asparagine. And the reason we do this is if you go back to the protein folding work in the 1990s from Buzz Baldwin's group, they showed that acetylation of any, in peptide models, if you acetylate any N-terminal residue, it enhances the helicity of that of the peptide, right? Asparagine had a similar effect as acetylation of a methionine in terms of the magnitude of the effect. And so we thought if this enhanced helicity is important, the asparagine at position 1 could behave similarly to acetylation, and we find that in fact it does. So we think it is, you know, what I want to say, it's some sort of structural stabilization that becomes important. And we know for disordered proteins, right, that when you undergo coupled folding and binding, that you know, sort of the structures that are sampled in solution may be important for recognition of partners. So I don't know how generalizable it is given that result, but I think it's a really interesting idea, and we certainly haven't explored it, but it's certainly an interesting idea.
Thank you. Thank you.
Okay, we have Alan Kosek. Can you ask the question? Following that, Long Cheng will ask. Liz, are you okay going up to?
Yeah, that's fine. Yep,
11:30 or
absolutely.
so? It's pretty informal now, so
Yeah.
yeah. Alan, go ahead. Alan?
Hi, can you hear me?
Yep.
Yes.
Thank you for the presentation, first of all. And my question is more like a general question about interactions of intrinsically disordered proteins, such as alpha-synuclein, that are playing key roles in degenerative diseases. And do you think this interaction is exclusive to alpha-synuclein, or can similar interactions or mechanisms involving lipids or sugar modified lipids be expected for these comparable other proteins as well?
That's a very good question, and I don't have a great answer for you. Like I will say, you know, I'm thinking there's a recent tau uptakes paper from Ken Kosik's group, right, where they looked at uptake of tau driven by LRRTM, which, you know, if it's a plasma membrane protein, it's probably glycosylated, right? So, but how broadly those specific glycan modifications are to recognition by the proteins binding them, I don't think has really been investigated in any meaningful way. I will tell you, we were planning to look at beta-synuclein just because that's kind of obvious, right? You know, it, you know, especially since a lot of the animal models showed that, you know, knocking out alpha-synuclein didn't seem to have major impacts on development. Knocking out all of the synucleins, you know, at least it was a little bit of a clearer phenotype. So we are curious whether or not beta-synuclein shows similar behavior in terms of especially the acetylation specific binding and recognition and glycan specific binding and recognition. So we were doing those now. We don't know the results of that yet.
Okay, thank you.
Are there any questions from the panel before going for the second round? Because
Yeah, I have a question.
John, Ari.
I have a couple of questions.
Go ahead.
First
go ahead.
of all,
Yeah.
great work, Liz. I really enjoyed it.
Thank
I learned a lot about alpha-synuclein, and it's interesting. I have a question regarding, so it binds and disrupts synaptic connections, and then it gets internalized. Which of the two, in your opinion, is damaging? Or in other words, is there a difference between alpha-synuclein before it leaves the cell, because it's intracellular usually, and when it comes back? Why is a trafficked re-entry of alpha-synuclein potentially cause damage? What could be what's the difference between the alpha-synuclein before and after it leaves the cell?
Yeah, that's a great question. And I have absolutely no answer for it, right? Like, you know, one idea that we have is that, you know, is that if you have more synuclein outside the cell, as you might in disease, if you have fibers being released or just, you know, overexpression of the protein as you see in this gene duplication, triplication, that you that maybe the normal regulation gets overwhelmed if you're driving too much Neuroxin off the surface. Yeah, other than that, I don't want to I guess get too far-fetched in my explanations. I honestly, I don't know. We've been trying to think of clever ways to follow synuclein sort of under normal secreted from a neuron and follow its pathway right back as it gets secreted and then re-internalized to see what we can learn about the protein. And we have some ideas about that, but nothing that's come to fruition yet. So I mean, the other thing is, right, is that maybe you have modifications that occur. Oh, I guess
but this would be
modifications that occur in aging or disease that then change what this interaction looks like, right? So one thing we haven't looked at all, or we have looked at, but the data is too preliminary, is disease mutants as well as modifications linked to disease besides the acetylation. Yeah.
I have another question. Do you know what normally what is the concentration of extracellular, you know, in the CNS fluid? What is the concentration of alpha-synuclein? Is it, because I know for A beta it's very low, it's in a sub-nanomolar.
Yeah, so I think it's a little bit higher for synuclein, although there may be someone on the call that knows this better than I do.
I'm asking because I noticed that the binding to Neuroxin is in the tens of micromolar the KD, something like that.
No, it's
20?
not. It is closer to. So that was just the only the binding. We just did sort of quantitative binding curves on and the hex cells expressing Neuroxin. Where we're increasing the amount of alpha-synuclein. I want to say it's in the tens of nanomolar. It's fairly high. We're currently also, I mean, hopefully we'll have a much better biophysical answer to this very shortly. I showed you the crystal structures of the Neuroxin 1 beta extracellular domain. So we're currently expressing that in suspension hex cells so that we can purify it and then actually do some NMR and other biophysics on it. So we ought to have hopefully a much better quantitative assessment of that binding soon. Yeah.
Are there any questions from other panel members? John, Magda, Bikash?
I have a question. So it's a kind of regarding the truncated forms of alpha-synuclein, which also abandon in cells. Have you thought of checking how they will behave?
Yeah, so interestingly enough, like 1 to 100 is a little bit bananas. Like it gets internalized much faster than the full-length protein. And it also seems to, like I was talking earlier and answering someone else's question, it also seems to escape from endosomes much better than the full-length protein. Shorter fragments bind Neuroxin, but don't get internalized. So that's also pretty interesting. And actually those shorter ones, they seem to bind with comparable affinity. They just don't go in.
Thank you.
Joan and Bikash. Okay, Longcheng, you are next.
Yeah, so my question is, think about in the PD, LBD, or MSA background, actually alpha-synuclein pathology is really complicated, reflected by different strands of alpha-synuclein. And for this in vitro, mediated PHF seem like much simpler than what from the human brain. So my question, do you ever try isolated like fraction, like a subcell insoluble, or like an actual solid vesicle isolated from the human brain or human patient brain, like PD brain, to test the similar intake maximum show in this your system? Is any differences in regulation, or they're following similar maximum? That's my question.
Yeah, we have done nothing with brain-derived materials, so I cannot answer that. Yeah, I don't know.
Yeah, the other following the truncation question, the previous question is a lot of people to started with the role of the core region, and to show core region somehow can show higher seeding ability. My question, do you ever try this just core region, mediate fibro, whether they can be intaken by the cell or they cannot at all? Thank you.
I don't know this. We haven't done that ourselves. Yeah, so if the N-terminus is missing, we would at least hypothesize that this uptake mechanism that I've been describing is no longer present. But as I said, like we certainly, especially in the case of fibers, can't rule out secondary pathways in terms of internalization. And I have no doubt that there are other pathways that can be implicated. And so if it's recognition of the core of the fiber that's important for uptake, then my assumption is that would be different than the processes we're talking about here. But we haven't done that.
Okay, thank you so much.
Wang, and then Bin, and then Anup, and we'll be done.
I don't have a question. Okay,
Bin.
Yes, hi Les. I have two quick technical questions. So I thought you said your N-terminal acetylated synuclein is biologically synthetic. It's not chemically synthesized.
Yeah, you co-express with so Dan Mulvihill in England basically has made a plasmid. It's available on Adgene, where it has the two components of the yeast N-terminal acetylase B in it. And we co-express that with synuclein and E. coli, and it goes to 100% acetylation. It's a really beautiful, easy way to do it.
Yeah, it sounds like magic. So does that system also how can that be like so specific to N-terminal? Do you think that will also accelerate other places?
So that's a really interesting question that I would have 3 weeks ago would have said, no, it only acetylates the N-terminus. I would say it's really specific in its recognition, I would say, right? So, it's not only that internal methionine, but it's the flanking residues that are important. And for example, if you took a NAT-C and co-expressed it with synuclein, it should not acetylate it because it doesn't have the right recognition sequence. That being said, when you're overexpressing a protein in E. coli, the mechanisms of regulation are going to be different or absent, and it's possible that you would sort of get some spurious modifications that you wouldn't normally get in a mammalian system. I will say that for the NAT-BS nucleon system, right, we mass spec all our samples, right, and there's never evidence for anything more than that single N-terminal acetylase on synuclein, but I don't know that you couldn't find conditions where it also modifies side chains, but in general, the N-terminal acetases are very specific in terms of their substrates.
Yeah, sounds a great system for you. I thought it was like an expanding genetic code kind of a system.
No, no, this is much easier than that.
Yeah, the other technical question I think you briefly touch on is you made the fibrillar alpha-synuclein you compared to the monomer about when they try to treat different cells, and you quote like I think Virginia Lee's method or something like that.
Yeah.
So how do you actually manage because I think the cell culture medium is very different normally versus like the buffer you promote for like fibrillar formation.
Yeah, absolutely, right? So, I mean, I think that's one of the things that confounds the amyloid field, right? Is depending on what buffer and what salt and how fast you shake, you get something different in terms of what you get when you aggregate, right? So I mean, the best that we can do is be consistent from sample to sample. Is how we aggregate. So at least we're within our lab comparing apples to apples. That doesn't mean we can compare our apples with anybody else's oranges, right? So I know, we haven't tried making aggregates under other conditions. I mean, sort of as Rams asked early on, we haven't tried making oligomers or any kind of, sort of something that doesn't go onto fiber and testing those. But that's certainly an interesting avenue for experiment. We just haven't done anything with that.
Yeah, I think you certainly have a very nice control there, but I think in the time scale of cell culture, those fiber may change in the culture medium.
It is definitely possible, right? It's a good point.
Thank you.
Thank you.
Okay, we'll take the last question. Anup, go ahead.
Hi, Liz. It was a good talk. I liked it. So I just had one question regarding the seeding ability of the acetylated form. You mentioned somewhere in the talk that it's probably not the best seed to promote cross-seeding or self-seeding, causing endogenous synuclein to aggregate or something. Do you think is it due to some kind of structural change, such as helicity imparted upon?
Oh, so I guess what I said is that the in vitro experiments, which is not our work, but other people's work, comparing the kinetics of the acylated unmodified protein, the acylated protein is slightly slower in terms of its aggregation. But if we think about the uptake experiments and the seeding of aggregation that occurs in those, it's actually the unmodified protein is having to seed acylated protein, right? Because that's what's present in the hippocampal cells, whereas the acylated protein is seeding like protein. And so that's actually a question I probably had thought enough about, and I don't know what's been done in terms of looking at cross-seeding between acylated and unacylated samples in vitro. But that's another possible explanation for why the acylated protein is so much better at seeding aggregation, is because it's seeding like material. And if there is a barrier to seeding of the unmodified material seeding the acylated material, that could also explain why aggregation is slower. It's a good point. Yeah, we haven't looked at that, but it's actually something we should look at.
Thanks.
Okay, thank you very much, Liz. It was a great talk and great Q&A session, and we learned a lot. It's a lot of new things. I learned a lot here.
Yes, I'm so delighted to see so many people here. So thank you to Ari and Duncan and well, I guess Ari's the only one left, but I'll send others emails to tell them thank you for.
Gilad left, but it was fun having all of them.
It was fun.
It's fantastic to see them.
Yeah.
All right.
Great
Thank
to see
you very much. Yeah, I really enjoyed it. It was really inspiring. Thank you for coming.
So much. Yeah, thanks for the invitation.
Yeah, nice meeting you.
Yeah, nice meeting you too. Bye-bye.
Bye.
Bye-bye.
Bye.
If everybody comes back on next Saturday, Ari, you should come back.
I'll try.
This is anti-aging process.
If you send me the link, you never send me the link.
The same link. Use the same link.
Okay, but that's the first time I got it.
We cannot bring Liz for every Saturday and she will kill me. Liz, Ari is very bad. It comes only for Liz seminar.
Yeah. Liz, say hi to my friend Abe Nitzan.
I certainly will. All right.
Yeah.
Take care, Ari.
Okay.
Good to see you. Bye-bye.
Bye. Nice seeing you. Thank you so much.
Thank you.